Hello everyone, Assalamu alaikum. First of all, I would like to congratulate the whole organizing committee of International Brain and Biomedicine Conference, especially the chief organizer, Dr. Ghulam Hussain, and his colleagues for conducting such a successful event. That is really a very golden opportunity, especially for the young researchers and students who get a chance to come across a lot of resources that could be really very helpful for their career. My name is Shamaila Zafar, and I am a postdoctoral researcher at Thomas Jefferson University. Here, I am working in the Department of Pathology and Genomic Medicine at Thomas Jefferson University. Before starting, I would like to share with you that my PhD is from GCUF Department of Physiology, and my PI was Dr. Ghulam Hussain. That's that's really very a blessing for me. So, well, currently my research focuses are on exploring the certain behavioral modifications and associated molecular underpinnings in developing brain under the influence of multiple factors, including neurotoxicants and the impact of the role of gangliosides in the pathophysiology of certain neurodegenerative conditions. So, thank you so much for inviting me and for giving me the chance to share with you about my research, and it's really a blessing to be part of this conference. So here I would like to highlight the impact of early life lead exposure on brain development and its reversal by environmental enrichment. So first of all, I would focus on certain influencers of the brain development, and then I will talk about some of my study design and share with you the summary of the findings. There are several factors that uh, significantly impact the brain development. These multiple factors contribute to the optimal neurodevelopment, and especially the exposure to the neurotoxicants, especially the lead. It has a capability to derail the neural and cognitive behavioral development. The other things that are present in our environment are our socioeconomic status, social behaviors, the richness of postnatal environment. They all can influence the offspring's brain behavior and social development. First of all, I would highlight how the lead how the offspring get exposed to the high lead concentration. So there are so many sources through which our offsprings get exposed to the lead, like drinking water, uh, soil, especially for the people living in the old homes and old buildings, because it still have the lead based paint. People living in urban areas, especially living near the highways, they have very high lead concentration in the soil due to the gasoline. So overall, it's really inevitable to avoid the lead exposure, and its impact in lead impact the whole body parts, including the brain. Uh, One thing that I want to mention here, that lead has nothing to do in our body. So lead in any concentration in the blood is detrimental for the normal development. Now, in the current scenarios, scientists agree that the lead concentration, even less than 10 microgram, like between 5 to 10 microgram per deciliter, uh, is still detrimental for the neurocognitive functions. Like the kids having the concentration between 5 to 10 have high, like less significantly decreased IQ levels. So they have, with aggressive behavior and irritability, high chances of aggression, and like in short, they really show the abnormal behavioral modifications in kids exposed to having a high lead concentration. Another factor that I already discussed is the socioeconomic status. A recent study has have found that there is an association between the income and the hippocampus. Hippocampus is the brain region that is involved in learning and memory, and it gonna be significantly affected by environmental stressors. So they highlighted that the what social aspects that gonna be associated with low income are, it's the type and complexity of stimuli in the postnatal environment, quality of parenting behaviors. Living in poverty is often associated with reduced environmental stimulation and suboptimal parenting. So there is fairly strong link between the low socioeconomic status and an increased likelihood of lower quality parental care. The negative effect of low socioeconomic status on kids' behavior and brain development are at least in part related to the ineffective parenting. So, the findings of another study indicate that the effect of poverty on hippocampal volume can be mediated by caregiving support or hostility. Like if let this state that if we enhance the caregiving support to the kids, although they belong to the poor family, but they see it gonna improve their capability, neurocognitive capability. But the same thing with less caregivers, like with compromised caregiving, can enhance the can more deteriorate the neurocognitive functions. So the effect of poverty on hippocampal volume in healthy preschoolers can be mediated in large part by quality of caregiving support. It suggests that the public health efforts could be helpful to enhance the quality of early caregiving by low socioeconomic status parents. That could be beneficial effects on the child development. So in current scenario, what needs to be wholly known is how the interaction of these social behavior factors can modify, either positively or negatively, the influence of lead neurotoxicity, and to what extent they can enhance, what extent enhanced environmental factors like enrichment or non-enrichment ameliorate the effect of developmental lead exposure. So our next objective and next objective is, to what extent does the environmental factors and parental care act individually and interactively to potentially modify the effect of early developmental lead exposure. To get answer for these objectives, the animal model could be an ideal choice because it's very easy uh, to apply the multiple factors, and even at the same time we can vary the complexity of the stimuli that's present in the postnatal environment, as well as observe the differences in the quality of maternal care on cognitive or behavioral functioning, and on the effects in the frontal cortex and hippocampus, in even amygdala at the molecular level. So here I'll share with you some very small study design. We provided the different factors and different variables, and they have been, which have been grouped depending on their different factors, like high enrichment, low enrichment, exposed to lead exposure, and control with no lead exposure, and with high maternal care and with low maternal care. And then these different cognitive behavior studies were conducted, and at the end of and some animals were kept for the molecular stuff. So here I will share with I'm going to share with you some the results of the trace pair conditioning, that is a test to assess the associative memory. And here on the RNA sequencing data of CA1 region of hippocampus. Our results showed that the quality of maternal care and the complexity of the postnatal environment interact and modify the effect from the lead exposure. The data of trace pair conditioning showed that the animals who get exposed to high lead concentration, get exposed to high lead concentration at the early age, they have shown significantly normal cognitive behavior as compared to those only in those groups who have high maternal care and enriched environment. Similarly, the analysis of differentially exposed genes showed that the environmental enrichment modified the lead-induced altered transcriptome. So it's just a summary of our study design and the findings. So here we employed three kind of factors like lead exposure, maternal care, and environmental enrichment, like whether enriched or non-enriched. So it is seen that the animals with high maternal care, enriched environment, although they get exposed to the lead, but they have shown almost normal cognition and normal behavior as compared to the animals who get exposed to the lead having low maternal care and non-enriched environment. So early life behavioral experiences due to differences in maternal care may either positively or negatively interact with the developmental exposure to alter the trajectories of adult cognitive and behavioral outcomes. The additional influences of the quality of the postnatal environment, whether it's enriched or non-enriched, further has the ability to interact with the variables of lead exposure and quality of maternal care to further alter the trajectory of adult cognitive and the behavioral outcomes. So enrichment, it can be concluded that enrichment really provided post-weaning has capability to reverse the lead-induced transcriptional changes, and it improves the associative memory functions. So it is increasingly appreciated that early life lead experiences affect broad life course trajectories, at least in part through the modifications of transcriptional regulations. The current findings also suggest that the interaction between the lead exposure and environment might also result in significant transcriptional changes in the brain of lead-exposed children that could have a profound influence on neuropsychological and educational outcome, further underscoring the potential importance of early intervention and environmental enrichment, especially for low socioeconomic status lead-exposed children. So that's all about these are the few things that I've shared with you. I hope it's going to be interesting for you, and you are more than welcome to ask if you have any kind of query, any suggestion. You are more than welcome. At last, again, thank you so much for giving me a chance to interact with you, to share with you, and it's really very encouraging for me. Thank you so much. Allah Hafiz.